DNA, deoxyribonucleic acid, a molecular blueprint for a living thing. In 1968, Gregor Mendel came up with his work on pea plants. He discovered the fundamental laws of inheritance. He didn't discover these by studying human beings, but rather the pea plant. This took him 8 years, and he published his results in 1865. It wasn't until 1900, after the rediscovery of his laws, that his experimental results were actually understood. In 1869, Friedrich Miescher isolated nuclein, DNA with associated proteins from cell nuclei. He became to be the first scientist to actually identify DNA as a distinct molecule. Between 1869 and now, many new discoveries have been found in DNA from a large variety of scientists. Determining what genetic factors can do, what DNA is composed of, X-ray diffraction pictures, demonstrating that the bases are equal, the shape, discoveries of individual genes, and the Human Genome Project. DNA is located on the nucleus of all cells, tightly coiled inside the chromosomes, except for red blood cells and bacteria. The shape of DNA is a double helix, often described as a twisting ladder. James Watson and Francis Crick worked as a team to work out the DNA structure. In February 1953, they built a model that established the general structure. This double helix is composed of backbones of alternating sugar and phosphate. Attached to these sugars are nitrogenous bases. There are 4 bases known as adenine, guanine, cytosine, and thymine. A and T pair together, which means C and G do as well. We have around 20 different kinds of amino acids in our body. You could think of them like building blocks. They live outside the nucleus in a place called the cytoplasm. Special chemicals inside the nucleus make partial copies of the DNA code. They are called RNA. The RNA travels to the ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, which tells the ribosome which chemicals to form, which makes a protein. Replication is another job the DNA does. In replication, the 2 strands are unzipped, and an exact copy is made by matching each complementary base. One old and one new strand are zipped together to provide duplication. Next-generation DNA sequencing has the potential to dramatically accelerate biological and biomedical research by enabling the comprehensive analysis of genomes, transcriptomes, and interactomes to become inexpensive, routine, and widespread, rather than requiring significant production. Several technologies have been developed more recently called next-generation sequencing that have sped up the process while reducing the cost. There are more and more technologies developing every day as scientists are growing more knowledge. DNA is very important as it carries our genetic information. Its discovery has not only revolutionized science and medicine, but has affected all walks of life. Thank you for watching.